Hello, and thanks for joining us for our latest webinar brought to you by Tecan, TagLite, two-cell assay, the next dimension to discovering new antibody drugs. I am Ashley, Managing Editor for Technology Networks, and I am here to moderate the event. Antibodies are central mediators of the immune system and, as such, are considered excellent candidates for the development of therapeutic drugs. As the complexity of these molecules increases, so does the demand for more rigorous assays to study them. This webinar will describe a new two-cell binding assay developed by Roche Glycart based on the TagLite assay from Cisbio Bioassays. This new assay system, established using Tecan's Infinite M1000 PRO microplate reader, opens up a new and exciting approach to studying protein-protein interactions in the drug discovery process. I'm pleased to say that we have two presenters for today's event. The first part of the webinar will be given by Sandra Grail-Richards, Senior Scientist and Team Leader at Roche Glycart, who will focus on the TagLite technology and applications with Tecan's Infinite M1000 PRO microplate reader. Sandra will then hand over to Michael Vital, Market Manager for Tecan Austria. Michael will be discussing customer issues encountered when doing cell-based assays and present key solutions and applications. So it's a warm welcome to both of you there. So just a little bit of housekeeping as well. So following the webinar, we will have a Q&A session, and obviously we're going to welcome any questions that you may have. So to ask a question, you should click the Ask a Question button that appears below the media player. Then simply enter your email address and question and click OK. We will answer as many questions as possible during the time available. Any questions we don't have time to get to, we'll be sure that you're contacted offline with an answer. Also, if you experience any technical issues, please use this button to request support. So I will now hand over to Michael, who will provide a brief overview of the agenda.
Okay, thank you, Ashley, for the introduction. I would like to welcome you all to this webinar to really look at a new two-cell assay using the TagLite assay. It's really an excellent solution for new antibody drugs. And without further introduction, I'm going to briefly go over the introduction for this webinar. So the TagLite assay will be presented by Sandra Grail-Richards from Roche Glycart. She will talk about the TagLite assay and will present how Roche Glycart actually established a new two-cell assay for new antibody drugs. And she certainly will present results, and then I will continue with a broad view of cell-based assays in terms of what problems customers usually face when they're going into cell-based assays, and it presents some solutions for the customers, talking about the Infinite M1000 PRO in more detail, and of course having conclusions and information and links for applications as well as webinar informations. So without further ado, I would like to hand over to Sandra.
Hello, so also from my side, thank you very much for the introduction and welcome again to today's webinar, which is designed from my side to present the new TagLite two-cell assay and its potential for the screening and characterization of new antibody-based drugs. My talk will be divided into 7 different parts. First of all, I would like to give you an introduction about TRFRET and the TagLite technology and its applications. Then I will go into more details about the two-cell assay, and therefore we use the human huFcγRIIIa IgG interaction as a model system. I will show you the assay setup and of course the results. And finally, I will give you an outlook about the potential of this new assay. So what is FRET? FRET by definition is the fluorescence resonance energy transfer, and it simply means the energy transfer from a donor to a so-called acceptor fluorophore. If you want to study protein-protein interaction, you can conjugate one of the proteins of interest shown in orange with the donor fluorophore and the other protein shown in blue with the acceptor fluorophore. You mix both proteins. If they interact, then you will be able to, upon excitation of the donor fluorophore, get an energy transfer to the acceptor fluorophore and measure its emission at a certain wavelength. This is shown in red in the bottom part of the picture. If the proteins do not interact, so there's no possibility for the FRET to happen. You will just be able to see the emission of the donor fluorophore shown in the upper part of the picture. For the FRET to happen, there are certain requirements that have to be fulfilled. First of all, the spectrum of the donor, the emission spectrum, has to overlap with the excitation spectrum of the acceptor. This is shown in the little picture, so you have first in blue the donor fluorescence and then the acceptor absorption. The filled blue area is the degree of overlap called J. Secondly, the distance is a really crucial mean in the FRET, because the fluorophores have to be really in close enough proximity for the energy transfer to happen. There's a term called Foster's distance, and this is the distance at which the energy transfer is around 50%. And in general, this is a bit smaller than 10 nanometers. Finally, the dipole orientation of the donor and the acceptor should be around parallel. The disadvantage of the classical FRET is that you often have a high background fluorescence. To overcome this problem, the TIA FRET, called time-resolved FRET, was introduced. This simply means that after the excitation, you have a certain delay time where you wait before then detect the specific FRET signal. In this delay time, you eliminate the short-lived fluorescence and therefore reduce the background. But to be able to measure in a time-resolved way, the donor fluorophores have to fulfill certain requirements. In general, lanthanides are used. These are rare earth elements like europium, terbium, or samarium, and they have a long emission half-life, about microseconds, which is important for the time-resolved measurement. They have a large Stokes shift. This is the distance between the peak height of the excitation and the emission of the fluorophore. They have sharp emission peaks and a high fluorescence. This is why, because they're in general complexed with organic moieties. In the upper picture, you see europium. In this case, it's cryptate, the organic moiety, and it acts like a cage that harvests the light and transfers to the fluorophore. So with this, I would like to start to introduce the TagLite technology. That is basically a FRET-based cell assay that was developed by Cisbio. The TagLite technology is a combination of the TIA FRET I just explained, or also called HTRF, and the SnapTag technology. Snap and ClipTag are DNA repair enzymes, and they are around 20 kilodalton in size, and the ClipTag is just a mutated version of the SnapTag. They are self-labeling tags and use benzylguanine and benzylcytosine respectively as substrates. There's a third tag available at Cisbio called Halo tag, and this uses chloralkane as a substrate. The Halo tag is a bit bigger, it's 27 kilodalton in size. So the advantage of the SnapTag we are generally using at Roche Glycart is that it is, as I mentioned before, a self-labeling tag. So once you have your protein fused with the tag expressed on the surface of the cell, you simply add the substrate. In this case, it would be the benzylguanine that is conjugated with the fluorophore, and the reaction just takes place, and within 1 hour your protein is labeled and the fluorophore is covalently attached to your protein of interest. So how can we prepare the cells? It's quite simple. You just have to clone your antigen or receptor of interest you would like to study on the cell surface into a vector with the SnapTag. You can have the SnapTag fused either at the N or C terminus of the protein, depending on which is extracellularly located. Then we usually use adherent Hek cells. We transfect them with lipofectamine. So we wait 20 to 24 hours, and then we add the substrate, and in a self-labeling mechanism, within 1 hour, the fluorophore gets attached to the protein of interest. It's a really easy assay, and after the labeling, you just detach the cells, you wash them, and then you freeze them in appropriate aliquots and store them at -80 degrees for your daily experiments. As we are usually only doing transient transfections, we do not put the cells back into culture after labeling. They go straight into the plates where we measure our experiments. So what are the applications of the normal TagLite assay? First of all, you could do an antibody competition. There you have your protein expressed on the surface that is labeled with the donor fluorophore. Then you add your antibody that is labeled with an acceptor fluorophore. You do a binding curve to find out the optimal concentration where you receive a really good FRET signal. Then you come along with the antibodies you want to test, and you have them in a different dilution, and if they bind the same epitope on the antigen, they will compete and you will see the decrease in the FRET signal. In general, instead of the labeled antibody, you could also think having a ligand there, and then you can search for antibodies that compete with the ligand disposition. Secondly, you can have a generic screening format. In this case, you have also the cell present there with the antigen labeled on the surface. Then you add your antibodies you would like to study, and you add a detection antibody that is labeled with the acceptor fluorophore. This will bind, and it can be an anti-human Fc, or it can be also an anti-tag antibody. And upon binding, you will see an increase in the FRET signal. At Roche Glycart, we generally use now this TagLite assay for certain and lots of different approaches. First of all, we do screening. This can be Fab or IgGs from supernatants, or also as purified proteins. There we check then just binding, non-binding. We also do EC50 and KD determination. And in a competition format, we do format evaluation. We can look at epitope binning when we want to compare different antibodies, and also search for ligand blocking antibodies. Quite new is now that we started setting up the simultaneous binding, and this is what I would like to talk to you about today. So as I mentioned before, we are using the human FcγRIIIa IgG interaction as a model system to set up the assay. FcγRIIIa, also called CD16A, is present mainly on natural killer cells and macrophages, and the interaction of this receptor with the Fc portion of the IgG leads to ADCC as effector function. So ADCC is antigen-dependent cell-mediated cytotoxicity. When you develop an antibody drug, it might be crucial that you don't have the ADCC. Therefore, then, you would start finding mutations or other alteration in the Fc portion to prevent the human FcγRIIIa interaction. In general, when we looked into these things, we just checked then afterwards the binding to the receptor, and if we couldn't see any binding, we said, okay, this is a real knockout. But then in 2012, we found a publication where they showed that when they had aglycosylated IgGs, which should not bind anymore to FcγR receptor, when they added the avidity effect by binding of the antibody to the antigen, they suddenly saw binding to the FcγR receptor again, although when they just checked the receptor site, no binding was observed. Then we thought, okay, we have to start to be really sure that we don't have an FcγRIIIa interaction. We have to start finding a way to mimic the avidity effect, and for the TagLite assay we thought, okay, we could introduce a second antigen expressing cells. Before I introduce then the assay, I would like to show you the alternative we used before we had the two-cell assay. So this was then an SPR-based assay, and we had the chip surface where we immobilized either the antigen, or here as an example, it's the receptor. Then we had as the first analyte the antibody that we wanted to measure, and then we added the antigens as a second analyte. So like this we could check the simultaneous binding. The advantage of SPR-based method, we use the BioCore, is that you can measure real-time kinetics, and you don't have to label the proteins. A disadvantage is that the proteins need to be purified, so you have to make sure that your antigen as well as the receptor is recombinantly expressed, and you can purify it in a way that it's structurally still functional. Also, the assay setup can be quite time-consuming, especially when you have problems with the surface stability. You also have to play around with injection times and concentrations. If the off rate, for example, also is too quick, then you have also a problem to detect the second interaction. And depending on the affinity, and this is often the case for the FcγR receptors, if you have a low affinity interaction, you have a quite high protein consumption. And high throughput is also not that easy. This is why then we came up with the idea of the two-cell assay. So how does this work and what do we expect to see? First of all, we have a cell, a Hek cell expressing human FcγRIIIa labeled with the terbium, the donor fluorophore. Then we introduce in the same well a second cell that is also a Hek cell transfected with the antigen and labeled with the acceptor fluorophore. The idea was now that if the antibody binds to the antigen and at the same time to the FcγR receptor, and we excite the donor fluorophore, do the cells come in close enough proximity that we have the energy transfer happening and we get the spread signal. The advantage of an assay like this would be that you don't have to purify the antigens or the receptor. They are present in their natural environment, so on the cell membrane. It's a really easy to use mix and read assay, no wash steps. You can have high throughput. We work in 384 well plates. And the protein consumption due to the low volume is also quite low. The disadvantage is you have to have the snap fusion on your proteins you want to study, and also you can't determine an on and off rate like you could in the biocore. So now I will go into more details about the assay setup as well as showing you the results. To set up the assay, we chose two antibodies. So they are basically the same antibodies recognizing the same antigen. The only difference is that on one hand we have the wild-type Fc portion, and on the other hand we have the FcγR receptor knockout. So we introduced mutations in the Fc portion shown as red stars that should prevent the binding to FcγR receptor RIIIa. For the two-cell assay, then it is important that we don't have any difference in the antigen binding, because otherwise we can't distinguish where the difference comes from if we see the curves are different or the binding is different. So first of all, we had to check both antibodies for the antigen binding. Then in a second assay, we just simply looked at FcγRIIIa interaction without having the antigen expressing cells. And in a third step, then we had the simultaneous binding. So first I will show you now the assay setup, then the results for the antigen binding. So we generated the cells, we transfected the Hek cells with the antigen Y fused to the snap tag, and we labeled it with the terbium. Then we used the antibody, so it's exactly the same antibody with the wild-type Fc. We labeled it with a kit from Cisbio. It's the acceptor, the 2 labeling kit, quite easy to use. And we labeled the antibody. We did a binding curve to find out the best concentration to start with the FRET signal. Then we added both antibodies, wild-type Fc or the mutated version, to the well in a concentration curve, and we checked for the competition. So we see a decrease in the FRET signal. We work in 384 well plates. The final volume per well is 20 microliter, and in this case we use 2,500 cells per well. The concentration for the labeled antibody, so this stays then constant in the well, is 0.77 nanomolar, and the concentration for the antibodies we wanted to check was from 700 to 0.02 nanomolar, the concentration curve. And we used the Tecan Infinite M1000 PRO reader to measure it. So the next slide will show you now the results of the antigen binding. So as I said, as it's a competition assay, we expect to see a decrease in the FRET signal if the antibodies we wanted to test are binding. And this is what we see. So you have in red the wild-type Fc antibody X, and in green, the FcγRIIIa knockout variant. And both, as you can see, both compete in a similar way, and also the EC50 values are quite similar, and this gave us the confidence that, okay, by introducing mutations in the Fc part, we do not alter the antigen binding. In the next step, we just looked at the FcγRIIIa interaction. It's the same principle, just in this case, we did not prepare the cells ourselves, but we purchased them from Cisbio. Cisbio offers a really broad range of human FcγR receptors as kits, so you obtain the cells expressing the receptor labeled with the terbium, the donor fluorophore, as well as in the kit included is a human IgG labeled with the D2, the acceptor fluorophore, and you set up the competition assay like I explained before. So we had the start point, we added again our both antibodies, the unlabeled ones with the wild-type Fc or the mutated version, and we had again the competition. Just this time it's not on the Fab arms, but it's on the Fc part, which is binding to the receptor. The setup is the same, 384 well plates, 20 microliter. In this case it's just 10,000 cells per well, and the concentration of the labeled antibody is 50 nanomolar, and of the unlabeled we went from 750 to 0.18 nanomolar. Again, we also here used the Tecan Infinite M1000 PRO reader for the measurement. On the next slide you see the results, and these look a bit different compared to the antigen binding, because of course here we expect that the wild-type Fc antibody competes because it should bind to the FcγR receptor and basically titrate out the labeled antibody that was there, but the FcγR receptor knockout variant with the mutated Fc should not bind. And exactly that is what we see here. So the green curve does not show any competition, and therefore we were sure, okay, this should be a real FcγR receptor preventing mutation. The EC50s you see below, it's a low affinity interaction, therefore it's around 150 nanomolar and no binding for the FcγRIIIa knockout antibody. But now, of course, we said this is good, but to be really sure that it does not interact with human FcγRIIIa, we introduce now the avidity effect by the antigen expressing cells. So we did the two-cell assay, as I explained really briefly before. We had the Hek cells purchased from Cisbio labeled with the donor fluorophore, and we had the Hek cells produced in-house with the antigen Y labeled now in this case with the acceptor fluorophore. If we add our antibody with the functional wild-type Fc, the antigen should be bound by the Fab arms, and then also the Fc should bind to the FcγR receptor, and if this happens, and we bring the cells in close enough proximity, the energy transfer should happen upon excitation of the donor fluorophore, and we should see a Fred signal. So with increasing the concentration of the antibody, we expect to see an increase in the Fred signal. Whereas when we add the antibody with the mutated version of the Fc, the antigen will still be bound, but the human FcγRIIIa should not be bound, and therefore when we excite the donor fluorophore, which is on the receptor part, we should not see any Fred. If the avidity effect is not somehow helping the Fc to bind again to the FcγR receptor. So then on the next slide you will also see the result here, and we were really, really pleased to see that it works, that even when you have two cells you can bring them in close enough proximity for the Fred to happen, and the binding curve is really nice. So for the wild-type Fc antibody X in red, you see a really nice binding curve with increasing the concentration, whereas the FcγRIIIa knockout variant does not show any binding at all. And this then gives us confidence that our mutation is a real mutation to prevent human FcγRIIIa interaction, and it also is a really nice control for the assay, because it shows that it's a real specific assay. So if you don't have the interaction with the receptor, then really you don't have any Fred signal whatsoever. So with this, I would like to summarize what I just showed you. So we believe that this TagLite 2-cell assay is a really valuable tool to evaluate multiple antibody interactions on living cells. Further, there is no need for the purification of your antigens, so if you have a really complex antigens, you don't have to worry, it will be just expressed on the cell surface, and therefore it's less artificial than an SPR analysis. It's also a really user-friendly mix and read assay, so you don't have any wash steps, which might be of importance when you study low-affinity interactions. And it's easy to scale down for low protein consumption. So with this, I would like to give you an outlook about what are we planning to do with this assay. So at the moment, we are extending the assay to other Fc receptors, including the neonatal Fc receptor FcRn. And what I can tell you so far is that it looks really well and it works pretty well, which is encouraging. Then we are also extending the assay to study simultaneous binding of bispecific constructs and the various formats of these bispecific constructs. This is important because these days the antibody drugs are not often simply antibodies anymore binding to one target, but they are binding various antigens, and it is a really important thing to characterize them in a way that you see can they both bind, both parts bind to the antigen, and also which format is the best one to use. So with this, I would like to move on to my last slide, which are the acknowledgements. At Roche Glycart, I would like to thank Pablo Mana, who's the head of Roche Glycart, as well as Claudia Ferrara and Christiane Jäger, who are my supervisors. And a big thank goes to Virginie Steinhardt. She's pipetting all of the TagLite assays, and she's always really enthusiastic, and I'm really grateful having her there. At Cisbio, it's Hamed, Delphine, and Maite. They support us with all the assay setups, and especially Delphine helped us a lot with setting up the 2-cell assay. And at Tecan, it's Roger, who's setting up the machines here in-house and does the training. Thomas, with him we wrote the application note about the 2-cell assay, and Michael, who organized the webinar. With this, I would like to thank you for your attention, and I'm very happy to answer any questions at the end of the session. Thank you very much.
Okay, thank you, Sandra, for handing over. Excellent presentation. We really are pleased to see how the assay has been set up. And as you mentioned now, what I would like to do is really step into a broader view in terms of cell-based assays in microplates. What have you actually seen at customer sites in terms of why are they stepping into cell-based assays, and more specifically, why should they actually go into cell-based assays? And of course, cell-based assays are in general more biologically relevant than simple biochemical assays because, of course, they predict more the in vivo situation, in example like drug sensitivity compared to an in vitro setting, of course. When you collect information on how drug candidates interact with cells early on, that helps you to help avoid late-stage failures in the drug discovery process, and of identifying crucial safety and efficacy data early in the process helps pharmaceutical companies, of course, to save lots of dollars annually in terms of their development process. In vivo modeling early in the preclinical stage is then driving really the demand for cell-based assays. And clearly what we see in terms of endpoint and kinetic processes, if you're looking at sort of time-dependent manner, this facilitates, of course, the investigation of biological processes. So what we see actually in terms of setting up cell-based assays at customer sites, we have identified at least 4 crucial points, what they are looking for and what they may not be aware of in general. So when you start cell-based assays, often, of course, you may want to use a microplate washer because obviously you're culturing cells in a microplate, and before reading, you want to remove phenol red before reading a plate, and of course, what we have seen in general, that many researchers still are using manual washing procedures with a simple pasture pipette and vacuum line, and clearly the problem could be, are you washing away your results? So next step in terms of the workflow, if you have your cell culture established, you want to protect your cells, of course, doing long-term live kinetic assays. And usually this requires the transfer of microplates between an incubator and the reader. And, of course, that poses the fact that the cells are often experience mechanical stresses and hostile environment, and, of course, that could change the cell in general. And if you then want to detect the cells in a microplate, you often may see that cells are not growing homogeneously, and so, of course, you may not get really consistent results when you do a readout in a microplate. And if you do long-term experiments in your laboratory, how do you actually know if your experiment is still running and if the cellular environment is stable without actually having the capability of checking that? So we are addressing these issues in terms of several really excellent solutions for the customers. For the first point, what we have done is really have implemented the so-called cell protection mode in the hydrospeed washer from Tecan, and did some very nice application studies looking at the comparison between manual washing and automated washing with the hydrospeed. And here we show basically two different cell types, sort of an adherent cell type and a weakly adherent cell type. And although sort of the wash efficiency simply is similar, when you then really look at the cell viability, so cell retention, then obviously it's a very big difference. So two key benefits for the customer: you get a lot more cells in your microplate, and obviously it's an automated process as well. If you continue with really detecting cells in an environment at constant conditions, then of course we're looking at the so-called gas control module, which Tecan has implemented and sort of really was the first company introducing this in a microplate reader, really meaning that you can now have the Infinite 200 PRO as the first multi-mode reader with adjustable gas concentration. So it's really a fusion between an incubator and a reader, checking the O2 and CO2 concentration simultaneously. And of course, looking at the data, you can see that, of course, with a constant readout in the reader, then with a gas control module, you don't have any data gaps, usually when you're swapping the plate between the reader and the incubator. When you detect cells, of course, then I mentioned non-homogeneous cell growth, and that could decrease your consistency of results, because clearly if you simply look at a microplate well in a microscope, then you may see that, of course, cells clump maybe on a corner and are not really homogeneously distributed inside a well. So how can you make sure you get consistent results? So the optimal read function implemented in the Tecan reader and the M1000 PRO, as well as the Infinite 200 PRO, is simply a function which means that when you consider, for instance, a normal fluorescence readout with, let's say, 20 to 30 flashes per read, that you make a simple pattern in the optimal read function as depicted here in 84, 96, and 384 well. And so these 30 flashes are then distributed across this pattern. So the actual time for the readouts is simply the same as for a normal readout, which you do when you read actually in the middle of the well. But as you can see, you get a much better consistency with the optimal read function, because the CV percent is much lower, particularly with higher cell numbers in the thousands. Now, the latest addition to our Magellan software version 0.2 is the so-called CNS software. So that comes back to the fact, the last point: how do you know if your experiment is still running, if the environment of your cells is still viable for your cells? And what we have done is have implemented a feature in the software which allows you to monitor your instrument actually outside the laboratory. So you can be at home, you can be in the office, checking your emails, but at the same time checking on the status of the experiment. And so this is implemented, of course, for all the Infinite readers, as well as for the liquid handlers, as well for the Evo. series, and this gives you a nice process security as well as the capability of really calling into the lab. Let's say if you have a M200 with the gas control module and you're running low on oxygen, you can call in and say, hey, please change the gas tank because otherwise I have to redo my experiment. So what are the customer benefits? I mentioned the monitoring of the oxygen and CO2 concentration, the gas control module. In particular with the M1000 PRO, if there is an integrated stacker attached to the instrument, so you're doing a semi-automated intermediate screen, then of course you get also information about the stacker on the CNS software telling you if the instrument is still running and so forth. You get information about the cycle number when you do kinetic runs, if the run in general is paused or has been completed, and of course how long the measurement actually will still be during the run, and gives you general features like instrument power failure and simple online and offline status. And talking with customers, so we obviously have to deal with customers in the regulated environment, like in the pharmaceutical industry, where often they are not allowed to have an open internet connection. And so one issue is to establish an internet server in an organization which allows you to install the CNS software. So it can be on a PC, but of course for research units which have capability of full internet access, there's the CNS app available on the iPhones, on the iTunes Store, and you can download the app on an iPhone or iPad, and then you can track actually all the actions I mentioned on a mobile device. So really nice feature for tracking the experiment you're currently running and remotely. Now when we go back to the M1000 in terms of how this instrument is actually being used, I only wanted to point out some main features which are excellent for cell-based assays and assay development in terms of what features can be used and benefits does the instrument really provide for the customers. Of course, it has been validated for all the major assays which are used in the research and pharma world, and is really the workhorse in terms of flexibility, sensitivity, and speed for all demanding applications in life science and drug discovery. One issue with the M1000 PRO has been addressed in terms of high sensitivity, really providing a filter-like performance, is the so-called premium quad 4 monochromator that basically brings more light, and it uses stray light down to 2.10 to the minus 7. And it includes also an adjustable bandwidth, so 2.5 nanometers below 300 nanometers and 5 nanometers more than 300 nanometers. That really allows you to control the bandwidth and the amount of light falling onto a fluorophore. Particularly, this is important when you do multiplexing assays with, let's say, 2 or 3 different fluorophores, and the spectra of these fluorophores are close together. Here, you can really use the feature of an adjustable bandwidth and get excellent sensitivity. The C-focus function with included background correction optimizes actually for the plate height and assay volume in the eye control software. So automatically the best signal to noise and signal to blank setting will be established in the eye control, and the same holds true for the gain. So how the gain function is basically implemented in the eye control as such that an optimal gain will be detected automatically in your assay. Absorbance reading in the M1000 is, of course, a multi-channel readout, but more or less it's also capable of reading the nanoquant plate for low volumes. So this is an issue when you do DNA, RNA, protein quantification at low volumes. In that sense, 2 microliters per spot. What I'd like to point out is the fact that the nanoquant plate is really the only plate which does not need any path length calibration. So it's ready to use. You just simply can clean the plate in, let's say, in a washer, and you don't have to recalibrate the path length. It's actually always ready to use for your DNA and RNA quantification studies. A key feature what we have seen, what people often use with the M1000 PRO is the 3D scan. That's particularly important for when you're doing assay development, when you basically have unknown compounds and you don't know exactly where the excitation and emission maxima is in the spectrum. And obviously, if you have no 3D scanning capability, it takes a long time to scanning the excitation and emission. So with the 3D scan, it's a simultaneous and excitation and emissions scan in the M1000 Pro to have really a feel where the peaks on the excitation and emission side are. And this is done with a single run. When we come to fluorescence polarization, we have implemented 4 LEDs as an excitation source because obviously LEDs compared to flashlight provide more constant light, and liquid crystal technology basically gives you the idea of really having the feature of generating polarized light at high speed, and together with the flexible wavelength selection from 280 to 850 in actually 1 nanometer steps and the adjustable bandwidth, we have an excellent sensitivity below 2 millip standard deviation in fluorescence polarization. So excellent for fluorescence polarization assays, for instance, like the Hek studies, like a Hek predictor mode. When we gave, for instance, for Luminous Sense basically allows you to use the installed filter wheel, and in the filter wheel are ready-to-use filters installed, so there's no need for selecting any specific filters because all the filters for the most common luminescence assays, spread 1, 2, 3, chroma glow, and so forth, even alpha screen and alpha laser, have been already implemented in the M1000 Pro. I would like to mention the Luminous Sense scan capability of the M1000 Pro, particularly for the characterization of new luciferases and mutants of existing luciferases. What we have shown here nicely, when you can do both in top as well as bottom optics, reading the vanilla as well as the firefly luciferase spectrum with a minimum resolution of 1 nanometer. So it's a really great feature in terms of looking at assay development for new luciferases in Luminous Sense assays. In the M1000 Pro, alpha screen and alpha laser capability has been implemented as such that we use really a high-power laser diode light source. I really want to point out that the infrared sensor for well-specific temperature correction is an excellent feature, and it has been optimized for 384 as well as 1536 well plates, and allows you to read, let's say, a 384 plate in less than 2 minutes. If you look now at the temperature correction function in alpha screen in the M1000 Pro, clearly, if you look at uniformity, sensitivity, as well as linearity, there's a clear data that basically temperature correction leads to better results. You get a better CV percent, you get a higher detection limit, and of course a greater linearity when you basically use the temperature correction in the alpha screen mode. At the end, of course, if you want to do your assay, we want to make sure that the instrument is actually in a quality-controlled environment in terms of you want to check if the instrument's basic function are usable. You can basically document your IQ/OQ situation, and with the multi-check capability of the M1000 Pro, this has been actually taken care of. Even the QC protocols are actually laid down in the instruction for use. You can find these protocols also in the manual. And finally, of course, if you want to do automation and more higher throughput applications, an integrated solution, Tecan, of course, offers with the M1000 Pro also the Freedom Evo series liquid handler, which for seamless studies and automation in higher throughput needs. I would like to point out 3 success stories in that sense, where we basically can show that the M1000 Pro as well as the Evo liquid handler have been successfully used. For instance, in Japan, where they, in Osaka University's drug discovery incubation group, they have opened basically their doors really to researchers in Western Japan, providing access for higher throughput screening capabilities, including Tecan, Freedom Evo, and the M1000. one nice story we can actually point to in terms of looking at sort of animal studies really is unwinding the genetic clock, where we can actually show you can actually read transgenic Drosophila with the M1000 up over a time course up to 2 weeks, looking in the endogenous reporter of circadian clock. And basically what we have shown here, bioluminescence sense can actually be studied in a regular interval. So you can see the light and dark cycle and the gray cycle. The gray cycle, the circadian rhythm is basically suppressed. And if we switch back on to a regular light and dark cycle, the basic bioluminescence reporter genetic clock starts again ticking. Finally, even live zebrafish can be studied with the M1000 PRO. We have a nice story where we can show that at Georgia Health Science University, Dr. Jeff Mumm was actually able to utilize the high sensitivity M1000 PRO to look at reporter-based assays even in zebrafish. Okay, the last section I'd like to point you to is the application notes we have for the M1000 PRO, particularly today's webinar, as you've already seen, was the TagLite 2-cell assay, and this application note is also available on our website. So please go on the website to the M1000 PRO, have a check on the application notes, and they are available for free to take a look at. Further on, what we have come up with is the application guide for multimode readers. So this is a nice piece for our customers. It's simply a cookbook for the laboratory. It's an easy way to get really nice results in that sense that the application guide offers a cookbook, and it's a compendium of most common assays. And for instance, if I look at the next slide, then the GFP assay has been laid out, and this is the general view of the application guide. So it's an assay overview about the principle, about the major applications, and what format the assay is about. And of course, it offers for each reader. In that sense, I have laid out the settings for the M1000 PRO, but it's also available, of course, for all the other readers from Tecan, what specific settings really have to be used to get optimal results with this particular assay. So, to conclude this webinar, we clearly have now shown that Roche Glycart has established a new 2-cell assay to open the route for really discovering new antibody drugs. And this assay was established with Tecan's Infinite M1000 PRO and offers the following benefits to Roche Glycart and further on, of course, for other customers who are going into this action and want to establish their own assays. So it's a simultaneous evaluation of multiple antibody binding interaction between cells. It's really user-friendly mix and read format, and it's easy to scale down, of course, for low protein consumption. And of course, I hope I could show you some aspects in terms of Tecan's solution for customer benefits when you go into cell-based assays, what kind of problems and what solutions you can expect to get excellent results for your assay. At the end, I would like to point you to important links for you to take a look at, particularly, of course, detection for our readers. The monochromator technology is laid out in terms of a Quad-4 monochromator video, which gives you a view of the optical path, which was laid out in the Infinite M1000 PRO. The particular aspects and technological situations and resources for the gas control module, the hydrospeed washer, introduction about the CNS software, and of course the resources in terms of webinars. Tecan is obviously implementing always a yearly webinar strategy that can be found in the /reader family link, as well as in the /webinars link. There is a history of all the webinars Tecan has conducted over the last 1.5 years. So please take a look at. And now, of course, we all thank you for your attention. And this webinar is now open to take questions and ready for discussion. Thank you.
Well, thank you very much, Michael and Sandra. I'm sure you'll all agree that was a really interesting presentation, and there's some fantastic points raised. So, as I said at the beginning, we'll move on to a Q&A session now. So, as we've had a number of questions in, I'd like to jump onto this straight away. So, the first question we've had in is, does fusing of the SNAP tag lead to any problems regarding the binding or structure of the antigen?
So I could answer this, I think. So in the beginning, we were also worried about that when we started using the TagLite assay. But now we've cloned more than 16 antigens with the SNAP tag and did assays. And I have to say that so far we haven't really experienced any problems. So I would say it's not a big issue. You might find that it's a problem, but so far we haven't seen any problems with the SNAP tag.
Okay, excellent. Thank you. The next question we've had in is also about the SNAP tag. It's asking, can the SNAP tag also be fused intramolecularly as well?
Yeah, we tried this once because it's possible that you can't use the terminus of the protein to fuse the SNAP tag. So what we did once, we cloned it after the transmembrane domain and before the ectodomain of the protein. And the only thing I can say is that we could label it, but the protein was a bit tricky anyway, so afterwards we couldn't detect any binding of an antibody to it. But yeah, in general, I think you could also fuse it within the protein. Excellent.
Well, thank you, Sandra. The next question is regarding a Tecan product. Is the F500 Tecan reader compatible for this technology?
Okay, Ashley, thanks for the question. Yes, of course, the F500 is also validated for TF-RET and Cisbio technology, so clearly it's absolutely compatible with this technology, so no problem at all.
Excellent. And one final question that's just come in is, does the two-cell assay require cells to be confluent to allow proximity of the receptor and antigen?
So confluent is a bit difficult to say because the cells are in suspension when we put them in the well. The only thing I can say is that a good start point is always when you use 10,000 donor label cells and 20,000 acceptor label cells to start with. So it is a bit crowded because they will settle down in the well. But yeah, this is always a good start point. I mean, confluence is difficult because they are not really growing in the well anymore.
Okay, excellent. And, okay, I believe, let me just have a look. One question that has just literally rolled in here is the CNS software expensive?
Mm-hmm. Okay, that's a question I have to answer. So basically the CNS software is actually more or less coming for free because it's part of the Magellan software package version 7.2. So if customers actually out there have already a Magellan software version 7.x, then as far as I know, they can really upgrade for free. And of course, I urge customers with versions below 7 to upgrade to Magellan version 7.2. And then, of course, the CNS is always included.
Okay, excellent. And one last question I think we've just about got time for is, can I run a multiplex assay in the M1000 PRO? I think obviously another question for you here, Michael.
Yeah, okay. Thanks for the question. So basically, there's always a little bit of misconception in terms of the naming, so multiplexing or multi-labeling. So clearly, what that refers to is simply if you have multiple detection technologies, for instance, if you want to compare or consolidate an assay with optical density as well as fluorescence intensity, so you have absorbance and FI read, you can simply do this in the eye control software by simply dragging the particular stripes for the detection technology in the workflow, so it's easy to use, and you can certainly do a multiplexing in the eye control software, so no big deal.
Excellent. Well, I think that's pretty much all we've got time for with the questions. So for anyone that asked a question we didn't have a chance to get to, please rest assured we will contact you offline with a response. I'd just like to say, sort of on behalf of myself, thank you very much to Michael and Sandra for an excellent presentation and an excellent Q&A session as well. So thanks again for that. The final point really is to to say that the presentation itself will be made available as an on-demand webinar. So if you think of any of your colleagues or friends that might be interested, please feel free to share the link, and it's something I feel a lot of people can benefit from. So again, thank you for all attending. Thank you.
Okay. Thank you, and goodbye.
Goodbye. Bye-bye.